Welcome to the class, students. This is the class on theory of equations. In previous class, I have solved some problems on the relations between roots and coefficients. Don't forget the formulas. That is S1 is equal to minus P1. S1 is equal to minus P1. S2 is equal to P2. S3 is equal to minus P3 and so on. So basing on this, welcome to the class, students. This is the class on theory of equations. In previous class, I have solved some problems on the relations between roots and coefficients. Don't forget the formulas. That is S1 is equal to minus P1. S1 is equal to minus P1. S2 is equal to P2. S3 is equal to minus P3 and so on. So basing on these relations, we have several problems on the theory of equations. So look at this problem. I am going to solve some more problems on these relations between roots and coefficients. So look here. Let alpha, beta, gamma be the roots of the equation px cube plus px square plus qx plus r is equal to 0. Then find equation. So it is a sigma alpha square, second bit sigma 1 by alpha. 1 by 1 I am going to find. So listen what I am doing here. So solution. Given that alpha, beta and gamma are the roots of x cube plus px square plus qx plus r is equal to 0. So alpha, beta, gamma are the roots of this equation that we have given. Okay. Then here chudandi. So this is P1, P2, P3. P1, P2, P3. Okay na? First one. I am going to write the relations. So then the relations are first one. Write down the relations between the roots and coefficients. The relations are first one. Sum of values S1 is equal to minus P1. Minus P1 అంటే first coefficient. అంటే ఇది ఎప్పుడు 1 గానే ఉంటుంది. సో 1 గా లేకపోతే చెప్పాను ప్రీవియస్ క్లాస్లో we have to divide the equation by the quotient of x power n. So this is minus P1. Yes, S1. S2. What is S2? S2 అంటే రెండేళ్ళు తీసుకోవాలి. Alpha beta plus beta gamma plus gamma alpha is equal to alpha beta plus beta gamma plus gamma alpha. S2 is equal to P2. P2 అంటే ఇక్కడ Q ఉంది. Next number 2. S3. Alpha, beta, gamma is equal to S3 is equal to minus P3 minus R. This is S1 is equal to minus P1 formula. S1 అంటే sum of roots. ఒక్కొక్కటి తీసుకోండి, ఆల్ఫా ప్లస్ బీటా ప్లస్ గామా. ఓకే, S2 is equal to P2. S3 is equal to minus P2. This is P1, P2, P3. Okay. Okay. These are the relations. Okay. So now, first one, sigma alpha square. Sigma alpha square is equal to alpha square plus beta square plus gamma square. Okay. So we have to find the value of sigma alpha square. Sigma alpha square means this one. This is in terms of PQR. PQR అంటే coefficients అక్కడ చూడండి. So that is these are PQR are the coefficients. Alpha, beta, gamma are the roots. So we have, by using these three relations, we have to express this alpha square plus beta square plus gamma square in terms of PQR. Okay, PQR lo cheppalante PQR ante enti. So we have to express this alpha square plus beta square plus gamma square in terms of these three things. That is alpha plus beta plus gamma వీటిలో రాయాలి. సో అంటే మనకు తెలుసు remember the things which I have explained in the previous class. Alpha plus beta plus gamma whole square. Whole square అంటే ఏమొస్తుంది చెప్పండి? Alpha square plus beta square plus gamma square plus 2 alpha beta plus 2 beta gamma plus 2 gamma alpha. So వాటిని ఆ 3 terms ని సబ్ట్రాక్ట్ చేయండి. minus 2 alpha beta plus beta gamma plus gamma alpha. ఓకే, ఇక్కడ వచ్చేటువంటి 2ab 2bc 2ca టర్మ్స్ సబ్ట్రాక్ట్ చేశాను. సో ఏం మిగులుతాయి చెప్పండి? దీన్ని ఎక్స్పాండ్ చేసినప్పుడు ఆల్ఫా స్క్వేర్ ప్లస్ బీటా స్క్వేర్ ప్లస్ గామా స్క్వేర్. దట్స్ ఓకే. సో ఇప్పుడు ఇది ఆల్ఫా ప్లస్ బీటా ప్లస్ గామా. సో ఏం మైనస్ పి హోల్ స్క్వేర్ మైనస్ 2 ఆల్ఫా బీటా ప్లస్ బీటా గామా ప్లస్ గామా ఆల్ఫా ఈజ్ క్యూ. Okay, this is equal to minus q whole square is nothing but p square minus 2q. Okay, this is the first bit. Okay, second bit. What is second one? Sigma 1 by alpha. Sigma 1 by alpha. This is equal to 1 by alpha plus 1 by beta plus 1 by gamma. This is equal to, so take the LCM, alpha beta gamma. So what is this first element? So beta gamma plus gamma alpha plus alpha beta. Okay, alpha beta plus beta gamma plus gamma alpha. This is equal to alpha beta plus beta gamma plus gamma alpha. So what is that? Alpha beta plus beta gamma plus gamma alpha. This is second relation which will be, so q by alpha beta gamma is minus r. It is minus r. So what is the answer? Minus q by r. So this is the second bit. Okay. Next one, I am going to solve this one, third one, sigma alpha q. Okay. Sigma alpha q. Okay, చూడండి, look here. So third bit, sigma alpha q is equal to sigma open చేస్తే alpha q plus beta q plus gamma q. So what is the formula? A cube plus B cube plus C cube minus 3ABC, okay, is equal to minus 3ABC is equal to minus 3ABC is equal to alpha A plus B plus C. Okay, remember the formula. So that is E alpha plus beta plus gamma alpha square. Okay, so so listen, alpha we know that alpha cube plus beta cube plus gamma cube minus 3 alpha beta gamma a cube plus b cube plus c cube minus 3abc is equal to formula. Remember the formula. A plus b plus c, a square plus b square plus c square minus ab minus bc minus cb. Okay? Okay. So this is equal to alpha plus beta plus gamma and minus p. Okay? Alpha square plus beta square plus gamma square. Just now we find here the first one. This is alpha square plus beta square. From that previous this one, the value is just now I solved here. P square minus 2q. P square minus 2q. Minus ఇందులో మైనస్ కామన్ తీయండి. వీటిలో మైనస్ కామన్ తీస్తే ఆల్ఫా బీటా ప్లస్ బీటా గామా ప్లస్ గామా ఆల్ఫా. ఓకే. సో నౌ this minus what we need alpha cube plus beta cube plus gamma cube. So e minus 3 alpha beta gamma will go to RHS. Okay. Alpha cube plus beta cube plus gamma cube is equal to, okay, minus p into p square minus 2q minus what is this? Q. ఇది ఏమవుతుంది? ఇది అటు ఇటువైపు వచ్చేస్తే మైనస్ 3 మైనస్ ఫస్ట్ ఇటు వస్తే ప్లస్ 3 ఆల్ఫా బీటా గామా. ఓకేనా? Now very simple, this is equal to minus p into multiply, minus p multiply చేద్దాం. Okay, minus p into p square minus p cube minus 2q minus 2 minus 3q minus plus 3p cube plus 3 into alpha beta gamma is minus r minus. Okay, this is your answer. That is, what is your answer? Minus p cube plus 3p cube minus 3r minus p cube plus 3p cube minus 3r. That is your answer. Okay, you have to remember this formula. సో మీకు ఈ ఫార్ములాస్ గుర్తుంటేనే ఇక్కడ చేయగలుగుతామండి. చూడండి, సిగ్మా ఆల్ఫా క్యూబ్ ప్లస్ బీటా క్యూబ్ ప్లస్ గామా క్యూబ్. దీన్ని వీటిలో ఎక్స్ప్రెస్ చేయాలి. దట్ ఈస్ యూ హ్యావ్ టు ఎక్స్ప్రెస్ దిస్ సిగ్మా ఆల్ఫా క్యూబ్ ఇన్ టర్మ్స్ ఆఫ్ ఆల్ఫా ప్లస్ బీటా ప్లస్ గామా ఆర్ దీస్ థింగ్స్. సో అందుకని సో వీ నో దట్ ఫార్ములా సో ఇది వెరీ వెల్ నోన్ ఫార్ములా. సో దిస్ ఈజ్ మైనస్ పి. దీని వాల్యూ ఇక్కడ మనం ఆల్రెడీ ఇక్కడ చేశాను నెంబర్ 1లో, అందుకని అదే కాపీ చేశాను. మైనస్ దిస్ వన్, సో మైనస్ కామన్ తీసాను, మైనస్ 2. ఓకే, సో దిస్ ఇస్ మైనస్ 3 alpha beta gamma send to the RHS plus 3 alpha beta gamma, so that this one. Okay, so that is about third. Okay, next fourth way చేస్తాను. What is fourth one? This one, that is sigma beta square gamma square, that is equal to sigma expand changing. Expand చేసాను బీటా స్క్వేర్ గామా స్క్వేర్ ఏం లేదు. సో బీటా ఉంటే ఆల్ఫా ఉంటే బీటా రాయాలి, బీటా ఉంటే గామా రాయాలి, గామా ఉంటే ఆల్ఫా రాయాలి. సో రొటేట్ అవ్వాలి. సో సిగ్మా రన్స్ ఓవర్ ఆల్ఫా బీటా గామా, బీటా స్క్వేర్ గామా స్క్వేర్. ఇక్కడ బీటా ఉంది, నెక్స్ట్ గామా రాయాలి, గామా స్క్వేర్. ఇక్కడ గామా ఉంది, ఆల్ఫా రాయాలి. మళ్ళీ నెక్స్ట్ టర్మ్లో గామా కాస్తే ఆల్ఫా అవుతుంది, ఆల్ఫా కాస్తే బీటా, ఆల్ఫా బీటా, బీటా గామా, గామా ఆల్ఫా. ఓకే ఓకే. సో ఇది, సో దీన్ని మనసులో మీరు ఏ అనుకోండి. సో ఇలా రాస్తాను, బీటా గామా హోల్ స్క్వేర్ ప్లస్ గామా ఆల్ఫా హోల్ స్క్వేర్ ప్లస్ ఆల్ఫా బీటా హోల్ స్క్వేర్. సో దిస్ ఇస్ ఏ స్క్వేర్ ప్లస్ బి స్క్వేర్ ప్లస్ సి స్క్వేర్. ఇందాక రాసాం కదా, ఏ స్క్వేర్ ఇక్కడ రాసాం ఫస్ట్ వన్, ఆల్ఫా స్క్వేర్ ప్లస్ బీటా స్క్వేర్ ప్లస్ గామా స్క్వేర్ అన్నప్పుడు ఏం రాసాం, అది ఇక్కడ రాస్తాను చూడండి. a square plus b square plus c square is a plus b plus c whole square, a plus b plus c whole square minus 2ab minus 2bc minus 2ca minus 2 బయటికి తీసాను. ఏబి అంటే ఇది ఏ ఇది ఈ రెండు కూడా మల్టిప్లై చేస్తే ఆల్ఫా బీటా గామా స్క్వేర్ అవుతుంది, ఆల్ఫా బీటా గామా. సో వీటంటే ఆల్ఫా బీటా గామా బయటికి తీసేస్తే ఇంకేముంటుంది, గామా ప్లస్ మైనస్ 2bc, మైనస్ 2bc అంటే మైనస్ 2bc అంటే ఇదండి, ఆల్ఫా బీటా గామా వస్తే మళ్ళీ ఆల్ఫా వస్తుంది, ఆల్ఫా ప్లస్ బీటా. సో మైనస్ 2ab మైనస్ 2bc మైనస్ 2ca. ఓకే, వాటిలో 2ab 2bc 2ca వీటిలో ఆల్ఫా బీటా గామా బయటకు ఒకేసారి రాసేసాను. సో మీరు కావాలంటే నెమ్మదిగా డీటెయిల్గా రాసుకోండి. వన్స్ అగైన్ ఐ రిపీట్ ద టర్మ్, a square plus b square plus c square is equal to a plus b plus c whole square minus 2ab minus 2bc minus 2ca. That's all. So now apply the things. What do we have? This is alpha beta plus beta gamma plus gamma alpha is, what is this, q square minus 2 alpha beta gamma is minus r. This is alpha plus beta plus gamma, alpha plus beta plus gamma is minus q square minus the minus minus 2rt, q square minus 2rt. Okay, so this is fourth one, and fifth one is also very simple. I'll do it just that. Okay, this is the last one, fifth one. So దీన్ని ఎలా చేస్తాం, very simple. Look here, alpha plus beta into beta plus gamma into gamma plus alpha. What is alpha plus beta? I write alpha plus beta as minus p, minus p minus gamma. ఈ gamma number this kind. Okay, alpha plus beta is minus p minus gamma. Next, beta plus gamma minus p minus alpha, gamma plus alpha minus p minus beta, send beta length. So మొత్తం మైనస్ ఇంకో కామన్ thing, minus into minus, that is minus p plus alpha, p equal to minus of p into p, వీటిని అన్నింటినీ మల్టిప్లై చేస్తున్నానండి, p cube plus alpha plus beta plus gamma p square. Okay, p into p, p square into gamma, gamma p square. అలాగే alpha into p square, అలాగే beta into p into p, okay, p square. Next plus alpha beta plus beta gamma plus gamma alpha p plus alpha plus alpha beta gamma. Very simple. Minus 5 వచ్చేసిందండి, p cube plus alpha plus beta plus gamma p square plus ఇక్కడ మైనస్ లేవు కాబట్టి అన్ని ప్లస్ వస్తాయి. ఆల్ఫా బీటా ఇంటూ p, ఆల్ఫా బీటా p, బీటా గామా ఇంటూ p, గామా ఆల్ఫా ఇంటూ p, అన్ని మూడు మల్టిప్లై చేస్తే ఆల్ఫా. ఓకే, ఇఫ్ యూ వాంట్ ఇండివిజువల్గా మల్టిప్లై చేసుకోండి, దెన్ ఆల్సో యూ విల్ గెట్ ద సేమ్. మైనస్ p cube, what is this? Minus p plus minus p into p square plus this is q, so this is q into p plus ఇది alpha beta gamma is minus r. Okay, ఇక్కడ p cube minus p into p square minus p cube will get cancelled, so this is ఇక్కడ రాస్తున్నాను minus p cube minus p cube plus p cube minus r. ఈ రెండు క్యాన్సిల్ అయిపోతాయండి. నెక్స్ట్ మైనస్ లోపల తీసుకొస్తే r minus p cube. What is it? See here, minus of minus r, r minus p cube, r minus p cube. That will be one answer. So simple. Look here, alpha plus beta is from this relation minus p minus gamma, minus p minus gamma. Similarly, beta plus gamma is minus p minus alpha, like that. So minus అంతా కామన్ తీసిన తర్వాత ఇలా వస్తుంది. ఈ 3 బ్రాకెట్స్ ని ఓపెన్ చేసి మల్టిప్లై చేయండి, ఇలా వస్తుంది. Then alpha plus beta plus gamma is minus p, this is q and this is minus r. Okay, simplify. Okay, this is the problem. Okay, you have to very aware of the formulas of algebra, that is a plus b plus c whole square, a cube plus b cube plus c cube minus 3abc, like that. Okay, I come to the next problem. I come to the next problem. Form a polynomial equation of the lowest degree. Form the polynomial equation of lowest degree whose roots are 2 plus r minus root 3, 1 plus r minus 12. Form the polynomial equation of lowest degree with roots whose roots are. Okay, ఇక్కడ రూట్స్ ఎన్ని ఇచ్చారు చూడండి. సో హౌ మెనీ రూట్స్ ఆర్ దేర్? గివెన్ రూట్స్ ఆర్ 2 plus root 3, 2 minus root 3, 1 plus 2i, 1 minus 2i. We have 4 different roots. Okay, 4 roots. So what is the lowest degree of the polynomial equation? 4. Fourth degree equation has at most 4 roots. Okay. సో కాబట్టి మనకు ఫోర్త్ డిగ్రీ ఈక్వేషన్ వస్తుందండి. దాన్ని ఎలా కనుక్కుంటాము? ఇక్కడ చూడండి, 2 plus root 3 అనేది ఫర్ ఎగ్జాంపుల్ 2 plus root 3 రూట్ కాబట్టి ఓకే, x is equal to a అనేది రూట్ అయితే ఏమవుతుంది? x minus a is a factor of f of x. కాబట్టి x minus 2 plus root 3 is the factor of f of x. ఓకే, ఇది రూట్ అయినప్పుడు a అనేది రూట్ అయినప్పుడు x minus a అనేది ఫ్యాక్టర్ అవుతుంది. అలాగే 2 minus root 3 అయితే x minus 2 minus root 3 అది కూడా ఫ్యాక్టర్. అలాగే ఇక్కడ సో కాబట్టి we have the 4 factors of the given polynomial. ఓకే, ఏం చేస్తానండి? The polynomial equation whose roots are the polynomial equation whose roots are 2 plus r minus root 3 comma 1 plus r minus 2i is very simple. ఇందా చెప్పాను కదా, x minus 2 plus root 3. ఓకే, ఈ x 2 plus root 3 రూట్ కాబట్టి x minus 2 plus root 3 ఫ్యాక్టర్. ఓకే, a అనేది రూట్ అయితే x minus a is a factor. It is one factor of the polynomial. ఓకే, మళ్ళీ x minus ఇక్కడ 2 minus root 3 కూడా రూట్ కాబట్టి ఇది కూడా ఫ్యాక్టర్. అలాగే 1 plus 2i అంటే x minus 1 plus 2i. నెక్స్ట్ x minus 1 minus 2i. x minus 1 minus 2i is equal to 0. Okay. This implies, okay, x minus 2 minus root 3. ఎప్పుడైనా కూడా x is equal to a అనేది ఒకటి రూట్ అయితే x minus c is a factor of. సో ఇప్పుడు ఇక్కడ ఎన్ని ఉన్నాయి? 4 రూట్స్ ఇచ్చారు. సో 4 రూట్స్ అంటే 4 ఫ్యాక్టర్స్ రాస్తారు. సో దీస్ ఆర్ ఆల్ ఫ్యాక్టర్స్ ఆఫ్ ది గివెన్ పాలినామియల్. సో పాలినామియల్ ఈక్వేషన్ అన్నారు కాబట్టి ఈక్వల్ టు 0 రాస్తారు. దిస్ ఇస్ వన్ ఫ్యాక్టర్ ఇంటూ అనదర్ ఫ్యాక్టర్ ఇంటూ అనదర్. ఓకే, వెల్ ఓకే, ఓపెన్ దిస్ బ్రాకెట్. Now look here, x minus a minus b, a plus b, a minus b into a plus b. ఏమవుతుంది? a square minus b square, a minus b, a plus b, a square minus b square. ఓకే, అలాగే ఇక్కడ a minus b into a plus b, a square minus 2i whole square is equal to 0. So ఈ 4 ఫ్యాక్టర్స్ ని మల్టిప్లై చేస్తున్నారు. ఓకే, దిస్ ఇంప్లైస్. సో ఏం లేదు, ఈ లోపల ఎక్స్పాండ్ చేస్తున్నాను. ఎక్స్ స్క్వేర్ చూడండి, ఎక్స్ స్క్వేర్ మైనస్ 4x ప్లస్ 4 మైనస్ రూట్ 3 హోల్ స్క్వేర్ 3, ఎక్స్ స్క్వేర్ మైనస్ 2x ప్లస్ 1. ఇక్కడ చెప్పండి i స్క్వేర్ ఇస్ మైనస్ 1. ఓకే, minus minus plus 2 square into 4 is equal to 2 square is 4. My I square is minus 1. Minus minus plus. So very simple. X square minus 4x plus 1. X square minus 2x plus 5 is equal to 0. Okay. Multiply each term by term. So ఈ రెండు ఫ్యాక్టర్స్ బ్రాకెట్స్ మల్టిప్లై చేస్తే we get a polynomial equation of degree 4. సరే నేను చేస్తాను కూడా ఎక్స్ పవర్ 4 ఎక్స్ స్క్వేర్ ఇంటూ వెరీ సింపుల్ మైనస్ 2x క్యూబ్ ప్లస్ 5x స్క్వేర్ మైనస్ 4x క్యూబ్ plus 8x square minus 20x plus x square minus 2x plus 5 is equal to 0. So this simplify just now. X power 4 minus 6x cube 9 plus 14x square. Okay, minus 22x plus 5 is equal to 0. So that is your polynomial equation whose roots are this one. Okay, so roots ఇచ్చారు మనకి 4 రూట్స్ ఇచ్చారు కనిపిస్తూ ఉన్నాయి 2 plus root 3 2 minus root 3 1 plus 2i 1 minus 2i ఈ 4 కూడా రూట్స్గా ఉండేటువంటి ఈక్వేషన్ 4 రూట్స్ అంటే మనకు మినిమం పాలినామియల్ ఈక్వేషన్ డిగ్రీ ఎంత ఉంటుంది 4th సో క్వాడ్రాటిక్ ఈక్వేషన్ 2 క్యూబిక్ ఈక్వేషన్ రూట్స్ 3 సో దే మే బి ఈక్వల్ ఆర్ దే మే బి డిస్టింక్ట్ ఓకే సో అలాగా ఇది బై క్వాడ్రాటిక్ ఈక్వేషన్ దీనికి ఎన్ని రూట్స్ ఉంటాయి ఆల్మోస్ట్ 4 రూట్స్ ఉంటాయి 4 రూట్స్ ఆర్ లైక్ దిస్ ఓకే సో ఇప్పుడు మనకి రూట్స్ ఇచ్చి పాలినామియల్ ఈక్వేషన్ కనుక్కోమన్నప్పుడు ఇలా చేస్తున్నాం అబ్జర్వ్ సో ఇది రూట్ కాబట్టి ఇది మనసులో ఏ అనుకోండి ఏ రూట్ కాబట్టి ఎక్స్ మైనస్ ఏ ఫ్యాక్టర్ అలాగే బి రూట్ అయితే ఎక్స్ మైనస్ బి ఫ్యాక్టర్ అవుతుంది పాలినామియల్ ఇలా రాసుకుంటూ వచ్చాం ఓకే ఇక ముందు మనకి పాలినామియల్ ఈక్వేషన్ ఇది ఇస్తారు అప్పుడు దానికి రూట్స్ మనం కనుక్కోవాల్సి వస్తుంది సో దట్ ఈస్ ద మెయిన్ మోటో ఆఫ్ దిస్ చాప్టర్ ఓకే రూట్స్ ఇచ్చినప్పుడు పాలినామియల్ ఈక్వేషన్ కనుక్కున్నాం సో దిస్ ఇస్ వెరీ ఈజీ పాలినామియల్ ఈక్వేషన్ ఇచ్చినప్పుడు వీ హ్యావ్ టు ఫైండ్ ద రూట్స్ ఆఫ్ ది పాలినామియల్ ఈక్వేషన్ ఓకే see similar model problems are there in your textbook you can solve all those problems on your own see here so I'm going to the next problem. Some simple problems. If alpha, beta and gamma are the roots of 4x cube minus 6x square plus 7x plus 3 is equal to 0. If alpha, beta and gamma are the roots of this equation, then find the value of alpha, beta plus beta gamma plus gamma alpha. Okay? So these are also very simple. Some simple. These are all of 2 marks problems. Given equation is come to the solution. Given equation is 4x cube minus 6x square plus 7x plus 3 is equal to 8. ఎప్పుడు కూడా రిలేషన్స్ ఇవి ఆల్ఫా బీటా గామా ఏంటి రూట్స్ సో మనకి ఇది కావాలి ఆల్ఫా బీటా ప్లస్ బీటా గామా ప్లస్ గామా ఆల్ఫా సో ద వాట్ ఈస్ దట్ S2 am I right S2 S1 అంటే ఆల్ఫా ప్లస్ బీటా ప్లస్ గామా S3 అంటే ఆల్ఫా బీటా గామా ఓకే S2 కావాలి అంటే S2 is equal to P2 P2 కావాలంటే ఫస్ట్ ఏంటి రిలేషన్స్ బిట్వీన్ రూట్స్ అండ్ కోషన్స్ అప్లై చేయాలంటే first you have to make your polynomial equation as monic polynomial equation that means 4th by cheyandi both sides divide by 4 on those given equation is this. So ఇలా చేసిన తర్వాత, so in order to avoid your confusion, here I write here P1. What is P1? This is P1, P2, P3. P1 is minus 6 by 4. Okay, that is minus 3 by 2. P2 7 by 4. P3 3 by 4. Okay, మనకి ఏం కావాలి ఇప్పుడు రాస్తాను. If alpha, beta and gamma are the roots, then we know that S2 is equal to P2. Okay na? So what is S2? S2 చెప్పండి, taking 2 what you have? Alpha beta plus beta gamma plus gamma alpha is equal to P2. What is P2 here? ఇది P1, P2, P3. P2 is 7 by 4. That's all. So suppose if this problem is asked for 2 marks, S2 మీరు ఇలా P1, P2, P3 రాసుకొని S2 is equal to P2 ఫార్ములా ఇది రాస్తే 1 మార్క్ వస్తుంది. నెక్స్ట్ దీని వాల్యూ కనుక్కొని 7 by 4 రాస్తే ఇంకో మార్క్ వస్తుంది. ఓకే, సో I come to the next problem. No doubt. If minus 1, 2 and alpha are the roots of the equation, so equation look at the equation. Okay, if minus 1, 2 and alpha are the roots of the equation 2x cube plus x square minus 7x minus 6 equal to 0, then find alpha. Okay, find the value of alpha. Okay, minus 1 to alpha are the roots of this equation. So very simple, given ఇవి రూట్స్ అన్నప్పుడు ఇంకా ఇప్పట్లాగా రాస్తాను. గివెన్ ఈక్వేషన్ is 2x cube plus x square minus 7x minus 6 equal to 0. This implies, సో ఎప్పుడు కూడా ఏం చెప్పాను, మోనిక్ పాలినామియల్ గా రాసుకోవాలి. డివైడ్ బై 2. ఓకే, సో హియర్ P1, హియర్ చెప్పండి వాట్ ఈస్ P1? P1 is equal to 1 by 2. P2 minus 7 by 2. P3 minus 6 by 2 and minus 3. Okay, so what is the first relation? We know that S1 is equal to minus P1. What is S1? S1 sum of 4 roots. Sum of roots ఏమి ఇచ్చారు? minus 1 plus 2 plus alpha is equal to minus P1. P1 is half 1 by 2. So very simple. Minus 1 plus 2 is a plus 1. Plus 1 send that side. Minus 1 by 2 minus 1, that is minus 3 by 2. Alpha is equal to minus 3 by 2. So once again see, this is also important for 2 marks. minus 1, 2 and alpha are the roots of the equation. Given equation is cubic equation. దానికి 3 roots ఇచ్చారు. Then find the value of alpha. So given equation is ఇది చెప్పి తీసుకున్నాము. ఓకేనా? సో దీన్ని మోనిక్ పాలినామియల్ ఈక్వేషన్ గా ఇలా రాసాము. ఇందులో p1 p2 p3 రాసాము. we know that s1 is equal to minus p1. s1 అంటే sum of roots minus 1 alpha plus beta plus gamma, sum of our roots. Okay. Okay. నెక్స్ట్ సో ఇక్కడ అన్నోన్ ఇచ్చినప్పుడు ఆల్ఫా కనుక్కున్నాము. అలా కాకుండా ఇప్పుడు ఐ కమ్ టు ది నెక్స్ట్ ప్రాబ్లం. ఓకే, see here. On the roots of the equation, on the roots of the equation, so what is the equation? Let's see. Minus 2 plus 6 minus 2. Then find the value of find A. Okay. So look at your problem. If 1 minus 2 and 3 are the roots of the equation, look at the equation. X cube minus 2x square plus ax plus 6 equal to 0. Then find A. This is your unknown. A కనుక్కోవాలండి. ఇక్కడ రూట్స్ ఇందాకేమో రూట్స్లో అన్నోన్ ఇచ్చారు. ఆల్ఫా ఇస్తే ఆల్ఫా వాల్యూ కనుక్కున్నాము. ఇప్పుడు రూట్స్ ఇచ్చారు, అక్కడ ఏ వాల్యూ కనుక్కోవాలి. సో ఇట్స్ వెరీ సింపుల్. If 1 is very simple, as since 1 is a root of the equation, x cube minus 2x square plus ax plus 6 is equal to 0. Okay? ఎప్పుడైతే 1 is the root of this equation, then since we have 1 has to satisfy that equation. So 1 in ఇక్కడ substitute చేయండి. 1 cube minus 2 into 1 square plus a into 1 plus 6 is equal to 0. This implies 1 cube and 1 minus 2 plus a plus 6 is equal to 0. 6 plus 1, 7 minus 2, 5. 7 minus 2, 5. అంటే 5 plus a is equal to 0. This implies a is equal to minus 5. ఓకేనా? ఇలా చేయొచ్చు. లేదంటే మీకు ఇక్కడ చూడండి, ఇది p1, ఇది p2, ఇది p3. సో ఇక్కడ p2 ప్లేస్లో ఏ అన్నోన్ ఉంది. p2కు సంబంధించిన రిలేషన్ ఏంటి? s2 is equal to p2. అలా కూడా చేయొచ్చు. సో s2 is equal to p2. ఓకే, వాట్ ఈస్ యువర్ p2? a. s2 అంటే ఆల్ఫా బీటా ప్లస్ బీటా గామా ప్లస్ గామా ఆల్ఫా. చెప్పండి. 1 into minus 2 alpha beta plus beta gamma plus gamma alpha is equal to a. So minus 2 minus 6 plus 3 is equal to a. A is equal to minus 8 plus 3. Minus 8 plus 3 minus. Okay? So ఇలానే చేసుకోవచ్చు, ఇలానే చేసుకోవచ్చు. ఎలా చేసినా ఓకే. So 1 అనేది రూట్ కాబట్టి 1 has to satisfy this equation. Substitute 1 here so that you got a is equal to minus Come to the next problem. Find S1, S2, S3 and S4 of the equation. X power 4, 86x cubed, minus 16 plus plus 86x square minus 176 176x plus 105 is equal to 0. This is your equation. Find S1, S2, S3 and S4 of the equation. X power 4 minus 16x cube plus 86x square minus 176x. Okay, that is your minus 16. Okay. S1 అంటే ఏంటి? S1 అంటే sum of all roots. So ఏం రాస్తాను చూడు. Let alpha 1, alpha 2, alpha 3 and alpha 4 అది fourth degree equation అండి, by quadratic equation. దానికి 4 roots be the roots of the equation. So what is the equation? That equation, see here, x power minus 16x cubed plus 86x square plus minus 176x plus 105 is equal to 0. Be the roots of the equation. Okay, no? So now we have to write here. So for your clarity, I am writing here. Yes. What is P1? P1 is equal to minus. So P1, P2, P3, P4. P2, what is P2? 86. P3, minus 176. P4, 105. Okay, now? So మనకి ఏం కావాలి? S1, S2, S3, S4. First relation. What is the first relation? S1 is equal to minus P1. Okay, now? S1 అంటే sum of all roots are taken one at a time. That is alpha 1 plus alpha 2 plus alpha 3 plus alpha 4 is equal to minus P1. Minus P1 is minus 16. Minus P1 and 16. So this is S1. Okay, S1. Number 2. S2 is equal to P2. Am I right? What is S2? Taken 2 at a time. Sum of roots taken 2 at a time. Yes, what is S2? Alpha 1, alpha 2 plus alpha 2, alpha 3 plus alpha 3, alpha 4 plus alpha 1, alpha 3 plus alpha 1, alpha 4 plus alpha 2, alpha 4. So all possible products of alpha is taken 2 at a time. 4 2 సెలెక్ట్ చేసుకోమంటే 2 మొత్తం 6 కాంబినేషన్స్, 6 pairs, 6 జతలు వస్తాయండి. alpha 1, alpha 2, alpha 2, alpha 3, alpha 3, alpha 4 like that is equal to S2 is equal to P2. P2 is 86. Okay. Next number 3. S3 cover. So S3 కి సంబంధించిన ఫార్ములా S3 is minus P3. So what is S3? Taken 3 at a time. అది కూడా రాస్తాను. alpha 1, alpha 2, alpha alpha 3 plus alpha 2 alpha 3 alpha 4 plus alpha 1 alpha 2 alpha 4 plus alpha 1 alpha 3 alpha 4. మొత్తం 4 కాంబినేషన్స్ వస్తాయి. These are combinations. Alpha 1 alpha 2 alpha 3 alpha 2 alpha 3 alpha 4 like that. So this is అంటే ఒకదాన్ని డ్రాప్ చేయాలి. S3 అంటే 3 తీసుకోవాలి. మన దగ్గర ఎన్ని రూట్స్ ఉన్నాయి? 4 ఉన్నాయి. 4లో ఒకదాన్ని డ్రాప్ చేయాలి. ఇక్కడ చూడు ఆల్ఫా 4ని డ్రాప్ చేశాను. ఇక్కడ 1 ఇక్కడ 3 ఇక్కడ 2. ఓకే సార్. సో ఈజ్ ఈక్వల్ టు minus P3 and minus P3 is 176. Next fourth one. S4 is equal to P4. What is S4? 4 ఉన్నాయి. 4 తీసుకోండి. 4 రూట్స్ ఉన్నాయి. alpha 1 alpha 2 alpha 3 alpha 4 is equal to P4. P4 is 105. Okay. These are S1 S2 S3 S4. ఒక బై క్వాడ్రాటిక్ ఈక్వేషన్ కి S1 S2 S3 S4 ఎలా రాయాలి, వాటి వాల్యూస్ ఏంటని ఇక్కడ మీకు చూపించాను. S1 అంటే సమ్ ఆఫ్ ఆల్ రూట్స్. S2 అంటే 4 రూట్స్లో 2 సెలెక్ట్ చేసుకోవాలి. ఎన్ని టర్మ్స్ వచ్చాయి? మొత్తం 6 టర్మ్స్. అలాగే S3 is equal to minus 2. ఈ మైనస్ని మర్చిపోకూడదు మీరు. యూ షుడ్ బి వెరీ కేర్ఫుల్ హియర్. సో S3 అంటే 4 రూట్స్లో 3 రూట్స్ తీసుకోవాలి. 3 రూట్స్ అన్నప్పుడు ఒకదాన్ని డ్రాప్ చేయాలి. ఓకే, మొత్తం 4 వస్తాయి. ఓకే, దిస్ ఈజ్ అబౌట్ దిస్ పార్ట్. నెక్స్ట్ there is a small problem here. Okay, I come to the next problem. If alpha, beta and 1 are the roots of the equation, look at this equation. So in previous problem there is only one unknown in the roots. It is 1 2 and alpha అని చెప్పాను. minus 1 2 and alpha like that. Here there are 2 unknowns. Then find alpha and beta. This is a see here, alpha, beta and 1 are the roots of the equation x cube minus 2x square minus 5x plus 6 equal to 0. Then find alpha and beta. Okay. This is also important for 2 marks. Since alpha, beta and 1 are the roots of the equation, x cube minus 2x square minus 5x plus 6 equal to 0. Okay, so then we have, since alpha, beta and 1 are the roots of this equation, we have, so I am going to write the relations between S1 is equal to minus P1, okay, implies ఇక్కడ P1, P2, P3 మామూలు. ఓకే, S1 అంటే ఏంటి? సమ్ ఆఫ్ ఆల్ రూట్స్. ఆల్ఫా ప్లస్ బీటా ప్లస్ 1 ఈజ్ ఈక్వల్ టు okay minus P1 minus P1 is minus 2. This implies alpha plus beta is equal to, this is 2 minus 1 1. Next, I come to the third relation. What is the third relation? S3 is equal to minus P3. So, second kuda teesukovachu, but I am taking this. What is third relation? Alpha, beta, gamma. Product S3 is equal to minus P3. S3 and alpha into beta into gamma. Okay, alpha, beta into 1. Alpha into beta into gamma. Okay, is equal to minus P3. Minus P3 is 6. This implies, student, S3 and alpha, beta, gamma. Alpha, beta, gamma and alpha, beta 1 are the roots. Alpha into beta into 1 is equal to minus P3. Okay. Alpha, beta is equal to minus 6. Okay. Alpha plus beta is 1. Alpha, beta is minus 6. How to find alpha and beta? Very simple. Product emo minus 6 ravali. Sum cheste 1 ravali. So this implies alpha plus beta is equal to 1 and alpha beta is equal to minus 6. Implies alpha is equal to 3. Beta is equal to minus 2. By guessing, that's all you can write. Don't write, there is no need to write that. So, if you rendu unnappudu alpha beta covenant em chestaru? Alpha minus beta whole square is equal to alpha plus beta whole square minus 4 alpha beta. Like that, ala rasi chestuntaru. So, not necessary. So, just by checking itself, you can write alpha is equal to 3, beta is equal to minus 6. If you want to check, substitute 3 minus 2, 1. 3 into minus 2, we got minus 6. Okay. So, we find the values of alpha and beta here. Okay. This is also important for the exam. Okay. So, in this class, I solved many problems which are important for 2 marks. So, and other remaining problems which are given in your textbook, you can solve all those things. I will continue in the next class.
Hello students, I am Sirisha PGT Botany KGBV Buchaipeta. Today we are going to learn the last unit in botany, that is the plants, microbes and human welfare. In this unit we are having 2 chapters. The first chapter is the strategies for enhancement in food production and second one is the microbes in human welfare. Now, in this class, we are going to learn the strategies for enhancement in food production. Enhancement of food production is a major necessity with the ever-increasing population. If the population increases, the food quantity also should increase. So, to increase the food production or the enhancement for food production, some strategies are applied to it. Some strategies are nothing but some techniques are involved which are used and with these techniques the food production is becoming more. We need like this strategies are applied to the most economically important plants. That is, some plants are useful for crops, that is for food, and some plants are useful for medicines, and some plants are useful for many other products. So, to enhance this type of plants, some techniques are used. Some techniques like you already learned about the animal husbandry and plant breeding, and these have a major role in our efforts to increase the food production. Animal husbandry relates to the animals, and plant breeding, this is a technique which we are using for creating, for yielding more amount of food. New techniques like mutation breeding, tissue culture and rDNA techniques are going to play a vital role in further enhancing food production. That means several not only plant breeding, some other techniques are also used like mutation breeding, tissue culture, or DNA techniques like this. These all techniques or all the strategies are used only for enhancement in the food production. Now we will see what is this plant breeding and how the food production is increased by using this technique. Before learning plant breeding, let us know about an Indian famous scientist M.S. Swaminathan. Moncampu Sambasavan Swaminathan is an expertise in genetics and plant breeding. At IARI, that is Indian Agriculture Research Institute, research on cytogenetics and radiation enables Swaminathan and his team to develop the short duration, high yielding varieties of rice, including scented basmati. Everyone know the basmati rice which is used for the biryanis. This rice, this variety of rice is introduced by the M.S. Swaminathan and his team, and they developed this variety by learning the course like the cytogenetics and radiation, using the techniques in that, and they came to know about they did more research in this field, so they are able to create a very good variety and a high yielding variety of rice, that is and one example is the basmati. Swaminathan is also known for the development of the concept of crop cafeteria, crop scheduling, and genetic improvement of the yield and quality of crops. His way of research, the more research, his mainly include the genetic improvement of the yield. That is the crop which is the crop which is developed by him, the plants should have a genetically improved, and also the quality of the crops should be good. So his concentration on that research helped him for this. Swaminathan's collaboration with Norman Borlaug culminated in the Green Revolution through introduction of Mexican varieties of wheat in India. He has been honored with the Padma Bhushan and many prestigious awards for his works. This is all about our Indian famous scientist, that is M.S. Swaminathan. Because of his works only, we are having more enough amount of food, and we are able to export this food to the other countries also. So his works are really remarkable, and so he's been awarded as awarded with Padma Bhushan. Traditional farming can only yield a limited biomass as food for humans and animals. Earlier days, traditional farming we used to practice the traditional farming for getting food, but this is for up to a limited extent, that is it is not sufficient for the whole population. With the increase of population, the yield also should be more. Better management practices and increase in acreage can increase yield, but only to a limited extent. Yes, if there are more acres, the yield might be more, but it is also not sufficient for the increasing population. The plant breeding technology has helped increase yields to a large extent. So with the limited area, and we should consider even the area also. So in that area, the yield should be more. That is, no crop should have any disease or should die, and every plant in that crop should yield more grains. And like this, we can this yield should be more. If the yield is more with this crop, then we will have enough food production. Like this, some technologies like plant breeding technology has helped increase yields to a very large extent. This technique, plant breeding technique, also is also useful for increasing the yield. And Green Revolution made it possible for our country to meet the national requirements in food production, but also helped us to export it. As I told, the Green Revolution is nothing but the increase of the economic importance of plants yield, that is to increase more number of plants. This is called as the green revolution, which includes the enhancement of food, that is the growing more crops or yielding more crops, and this is so much of sufficient that we are able to export to other countries also. This is all the green revolution means. It is deals about the increase of the economic importance of the plants, and plant breeding techniques for development of high yielding and disease resistant varieties in wheat, rice, maize, etc. So these plant breeding techniques mainly for getting more yield. For getting more yield, we means for this is more suitable for the crops. That is the food crops like wheat, rice, maize. All these food crops we need the yield more. That is the grains should be more amount of grains. So if we get more amount of grains, this food will be sufficient for the population. So this plant breeding technique is very much useful for the crop plants. And now let us learn about the plant breeding. What is plant breeding? Plant breeding is the purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation, give better yields, and are disease resistant. Conventional plant breeding has been practiced for thousands of years. Many present-day crops are the result of domestication in ancient times. This is a type of plant breeding, and in this type of plant breeding, this is by means of the natural methods. By natural methods only, the plants they are selected and they are breeded. With this method is nothing but the conventional plant breeding. And second type is the classical plant breeding, and in this classical plant breeding involves crossing or hybridization. Hybridization, that is the plants are selected artificially and they are crossed, so that this type is called as the hybridization. And this hybridization of pure lines followed by artificial selection to produce plants with the desirable traits of higher yield, nutrition and resistance to diseases. So the plants which are produced by this hybridization, that is after this hybridization, the plants which are formed are generally the pure lines. They are pure lines means they are having the same genetic combination, the constitution. This same genetic that is it is called as the homozygous. This homozygosity of plants which is having the same genetic structure, it is mostly done by the artificial selection and they are crossed. This type of practice is called as the classical plant breeding type. And these plants which are obtained by this also have a higher yield, and also they are useful for the high nutrition and resistance to the diseases also. And that is they are protected from the diseases. With advancements in genetics, molecular biology and tissue culture, plant breeding is now increasingly being carried out by the using molecular genetic tools. So more advanced research done in the plants. Now these genetics or molecular biology or tissue culture, even the plant breeding is also used, and it is practiced and it is developed more by using some genetic tools also. The main list with the plant breeding deals with the increasing crop yield, increased tolerance from the stresses, and resistance to pathogens, tolerance to insect pests. That is plant breeding. The advantages, you can say the advantages of plant breeding is increasing in crop yield. I told the yield should be more, the more amount of food grains should be produced, and increase to tolerance from environmental stresses like whether it is hot or whether it is too hot or whether it is too cold, the plant should survive. This is called as the increase to tolerance from environmental issues. And resistance to pathogens. that is, it can tolerate even if it is affected by virus or bacteria or fungi like that. And tolerance to insect pests. We know in the crops, in every crop some pests will be there, some insects are there. So if the insects, they spoil the crops, so these plants should they should tolerate the from the insects. That is the with the plant breeding technique, this problem is also solved. That is the tolerance to insect pests. Coming to the steps in plant breeding, there are 5 steps which should be followed for the plant breeding. First one is the collection of variability. Second, evaluation and selection of parents. Third one, cross hybridization among the selected parents. And fourth one is the selection and testing of superior recombinants. And fifth one is the testing, release and commercialization of new cultivars. So all these steps we are going to learn in the detail now. The first step is the collection of variability. Genetic variability is the root of any breeding program. So we need the genetic changes of the plant. Suppose the plant is having very small seed grain. No, it doesn't work. So we want a grain which should little bit larger. So this genetic variability should be there for the breeding program. So generally, pre-existing genetic variability is available from the different wild varieties of crop plants. We have some local varieties of crop plants and also some wild varieties of crop plants. In that wild varieties, we will have some little bit, the grains may be more larger in size or the yield may be more. So these varieties, that is this, their genes are, or the genetic variability is there. That is the difference of genes is there from the local variety to the wild variety. So these type of plants we require. So collection and preservation of all different wild varieties, species and relatives of the cultivated species is a prerequisite for effective exploitation of natural genes available in the populations. So these varieties, these wild varieties, we have to collect, and we have to preserve them. And the entire collection of plants or seeds having all the diverse alleles of all genes in a given crop is called germplasm collection. So here we are collecting the plants or the seeds. We are collecting the plants or the seeds with the diverse alleles of genes. Means the composition of the genes having all the required genes. That is the quality of the genes are high, like we require a big size grain, and maybe we doesn't require more number of leaves, or like that. Whatever the qualities we require, there is a change in the genes. These change in the genes, or this is called the diverse alleles of all genes in a given crop, is called as the germplasm collection. So in the first step, we are collecting the different different types of genes present in the seeds or plants. And the next step is evaluation and selection of parents. The germplasm is evaluated so as to identify plants with desirable combination of characters. That is, the germplasm should be tested. That is, whether it is all the desirable characters are there or not. This should be evaluated. And then the selected plants are multiplied and used in the process of hybridization. So whatever the plants we are selected with a good quality, they are some characters are very good in that plant, those plants are selected and they are multiplied. That is, they are self-pollinated at the end. They are, and the hybrids produced from them are called as hybrids, and this process whole is called as the hybridization. And now the pure lines are created wherever desirable and possible. So that is, pure lines are nothing but the same homogeneous condition, that is, they are having same genetic combination. These are called as the pure lines. These are created whenever desirable and possible. Means we have that particular variety of a plant is very good at all characters. Let it is the yield is more for that plant. Those plants are only crossed and the pure lines are formed. They are created more number of plants, the same plant, genetically same plants, they are produced more in number. And the next step is the cross hybridization among the selected parents. The desired characters have often to be combined from two different plants. For example, high protein quality of one parent with a disease resistance of another plant. So, two types of plants are selected. One is very good at the protein and other one is can tolerate the disease. That is their disease resistant plant. So, in one plant we are having one quality, that is the food which is produced from that grain which is produced from that, it is in high protein quality, but it is not resistant to disease. But other one, the food grain may not be have a high protein, but the plant is disease resistant. So if these two plants are selected and if these two plants are crossed, then the next hybrid will be produced should have high protein quality and also it should be disease resistant. Means we should get the hybrid plants of both the desired qualities. Next, this is possible by cross hybridizing the two parents to produce hybrids that genetically combine the desired characters in one plant. As I said, that plant which is produced that is called as a hybrid, and this hybrid should have both the qualities of the parents. That is a parent plant of one is having high protein and other parent plant should have is a disease resistant. Though the hybrid should have both these qualities. That is only possible through cross hybridization. And next, however, it is not sure that the hybrids do not combine the desirable characters. Usually one in a few hundred to 1,000 crosses shows the desirable combination. It is not that easy method. We should cross so many crosses should be done among these parents, and then only about a ratio of 1 among 100 the plant can have both the qualities which we are looking for the desired qualities. This is the step number 3. And the fourth step is the selection and testing of superior recombinants. Selecting among the progeny of hybrids those plants that have the desired character combination. That is, whatever the characters we are looking in the plant, these are first selected. Then, selection process is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. This selection process is very important, and it is also required to test the qualities whether it is present or not. And next, as often many superior progeny plant may be available, and these are self-pollinated for several generations till they reach uniformity, that is homozygosity, so that the characters will not segregate in the progeny. So the desired qualities of the plants which we get after the cross hybridization, these plants are self-pollinated. Why self-pollinated? Means we will get the same desired genes for many generations, and thus the quality will be maintained, so that all the plants will have a same genetic constitution, and or you can say it is as the homozygosity. That is, these plants are only self-crossed and more number of yield is produced. This is the fourth step. And the next step is the testing, release and commercialization of new cultivators. The newly selected lines are evaluated for their yield and disease resistant. This evaluation is done by growing these plants in research fields. Testing of hybrid line is done by growing these plants in research fields. So after cross-pollination, the hybrids are produced, and these are also evaluated, and more research is done to these. And after testing these plants, the testing of hybrid line is done by growing these plants in research fields. So we show the so many generations, they will check these plants are maintaining the same homozygosity, and the variety is perfect. And this research, completely research of this hybrids are done in the research fields. The tested material is evaluated in comparison to the best available local crop cultivator. So these are these plants are tested, like they are compared how before variety and now the after the advanced now present variety. They will see the comparisons, and they will also test whether it is this plant producing grain does not have any side effects, or like that they will do the research. And then the release of tested material is done after selection and certification. Then the seeds produced by that plants, they are tested, and also they are selected and they are certified. So if they give certification that these seeds can be used, these seeds are only given to the farmers to proceed for cultivation. Then that is comes under the commercialization of the new cultivators. Now we will learn another technique, tissue culture. Till now we learned that the plants we are the desired qualities which we want for the plants, we are using the technique plant breeding. Now this is another technique called the tissue culture. Here we know there is no need of selection of parents or their seeds. Just we can collect the one plant which is which all the desirable characters are present in that plant. We select that plant, and in that plant we can take any part from the plant. That is, we can take any part means we can take any cell or tissue or any organ from that plant, and we can multiply into many plants. That is what is happening in the tissue culture. So this is one technique where here there is the culturing of the cells of the desired plant, the cells or the tissues or the organs. We take a small amount of the cells or tissues, and we grow into them into number of plantlets. This is happening in the tissue culture. That is this technique. The technique of growing, culturing and maintaining cells, tissues and organs in vitro is known as tissue culture. Here we are not doing any hybridization or artificial hybridization or nothing. We are just culturing those parts in the lab. In the laboratories we do this. So this technique is called as the tissue culture. It is based on the cellular totipotency. The totipotency means the capacity to generate a whole plant from any cell is called the totipotency. Totipotency means the ability of the cell, that part of the cell can regenerate into a whole new plant. That is called as the totipotency. The capacity to generate into a whole plant from any cell, that is called as the totipotency. Now let us learn the techniques or the steps involved in the tissue culture. First one is the preparation of nutrient culture medium. Second, the sterilization of the culture medium. Third one, preparation of explant. Next, inoculation of explant. Next one is the incubation for growth, and last step is the acclimatization of plantlets and transfer to pots. Now we will study each step in detail now. Now let us see the steps involved in the tissue culture. Preparation of nutrient medium. So the nutrient medium must provide a carbo source and also inorganic salts, vitamins, amino acids and growth regulators like auxins, cytokinins, etc. So as we said, the cells should grow in a medium. Because we are taking not a plant or a seed, we are taking small amount of cells. So these cells should be provided, means they should be kept in a medium. Medium is nothing but a quantity which is made with all auxins or jelly like substance. You can say like it will be like some jelly like substance, a small gel. If you take in that jelly like substance, all these vitamins, amino acids, whatever the plant requires to grow, all these are provided. It is very rich with vitamins, amino acids, inorganic salts, and also the growth regulator. The cells should grow, so growth regulators also should be there, like auxins, cytokinins, etc. This nutrient medium, that is why this is called as a nutrient medium. Nutrient medium is this medium, the food, we can say this is like it is nothing but like a food, small baby providing with all good food, like we provide all the medium. The medium is nothing but a small jelly like substance, which in this all the important minerals, salts, and auxins, growth regulators, everything will be present. In this medium only the cells can grow. And now this medium, the sterilization of the culture medium. The culture medium is rich in nutrients and therefore attracts microorganisms. So the medium should be sterilized. Sterilization is carried out in an autoclave for 15 minutes and 121 degrees centigrade and 15 lb pressure. So here the medium, what I said, already we prepared one medium. This is very rich in nutrients. So what happens, all the microorganisms present in the atmosphere, there is a chance to grow in that medium, because all minerals, also vitamins, everything is present there. The microbes can happily grow in that medium. So these microorganisms should be avoided. The means it should not, that it should be sterilized. That sterilization of the medium should be done. Otherwise all these microorganisms will grow in the medium, and they will not allow the cells to grow. So it should be free from the bacteria and virus or other microorganism. So this medium should be sterilized. What we will do for sterilization generally? Sterilization we will do generally heat. Sometimes we do boiling, so that it will the microorganisms will die. Like that here also, that this medium is sterilized in an autoclave. An autoclave, it is a machine which clears, at 121 degrees centigrade and 15 lb pressure, if you keep that autoclave, these microorganisms, there is no chance of microorganisms to grow in the culture medium. So this process is called the sterilization of the culture medium. And the third one, then the third step, next step is what? Preparation of explant. So now we kept all the nutrient medium ready, and now in this medium we should make some these cells to grow in this medium. So to collect the cells, what we will do? That is preparation of explant. Any living part of the plant, such as roots, stems, etc., which is used as inoculum, is called explant. That is what you want to take from the plant, that is maybe from the root, maybe from the stem, or the cells what we want to take from the plant. These cells should be taken, that those cells which are taken from the plant is nothing but the explant. These plant, maybe the cell, maybe a tissue, this part is called as the explant. Explant means we are taking some a small piece of cells or small piece of tissues from the plant, and this explant is grown on the medium. This is called as an explant. So here what it is not like that we can take from any root tip, we can take from any part of the root or any stem. It is like we should take from only the tips of the root or stem, because auxins are more in the tips. Auxins, growth regulators are there. These auxins are more in the root tips and the shoot tips. That's why in tissue culture, the explants which we collect or which we use, we collect from the root tip or the shoot tip, so that more growth regulators are present at the ends, at the tips. These cells are collected, and this is called as the explant. And now what we will do with this explant? Next one is inoculation of explants. The transfer of explants onto the sterile medium is called inoculation. Now, the transfer, what we have collected the explant, now what we have to do, this explant should be on kept on the sterile medium. Already the nutrient medium which we prepared, that medium is ready, and now we are transferring the explants to the onto the sterile medium. This is nothing but the inoculation of the explants. It is carried out in the laminar airflow chamber. Because after sterilization also, and if we remove the medium and keep it again in the air again the microbes is present, again there is a chance of entering all the microbes into the medium again. So what we will do, this all this transfer of explant on the medium is done in a laminar airflow chamber. There will be a chamber. In that chamber is also very much sterilized chamber, and in that chamber there is no chance of entering of microorganisms. In that chamber only we will do the inoculation of explant. Now next is the incubation for growth. The cultures are incubated for 3 to 4 weeks. During this period, the cells of the explant absorbs nutrients, grow and undergo repeated mitotic divisions. They produce an undifferentiated mass of cells known as callus. So everything is fine. We have transferred the explant on the medium. Now what will happen? We have to wait in the we have to watch for 3 to 4 weeks daily that there is any improvement of the cells. The cells should continuously divide, because all nutrients are supplied. We are providing all the growth regulators, all the vitamins, everything we are providing in the medium. So cells should grow. The cell should multiply. It repeated mitotic divisions. The cells can easily multiply and it will grow in a mass of cells. This condition, this mass of cells is known as the callus. So if this is happening means it is a success. We are succeeded in that the cells are happily growing, and we are forming one mass of cells, and this mass of cells forms a tissue, and a group of tissues form an organ, and a group of organs form a complete plant. So this whole process should take place only in the lab, and that is the reason we have to observe these cultures only in the lab for 3 to 4 weeks. And next, auxins and cytokinins are added to the culture medium so that the callus is induced to produce organs like roots and shoots. Because natural plant will have its own auxins, and it takes water from the soil and it supplies. But here we should supply all the vitamins and minerals which were required for the callus to grow. That is, auxins and cytokinins are must for the growth. The auxins and cytokinins are the growth regulators. So if we keep more amount of these auxins or cytokinins, then this it will be very easy to these plantlets to callus to grow into plantlets. And this phenomenon is known as the organogenesis. Because from the callus it is forming till organs like roots and shoots also, like it is forming this process from the callus, that is from the cells, again callus from callus again tissues again forms organs, and from this whole process is called as the organogenesis. The explant develops an embryonic callus through embryogenesis, from which embryoids are produced. So here explant which you have taken can develops an embryonic callus. That is, all some small embryo is formed. This embryo also can be cultured. This is called as the embryogenesis. And from the embryo, the embryoids are formed. The embryoids which consist of small roots and shoots. Since these embryos develop from somatic tissues, they are referred to as embryos. So here these somatic tissues are nothing but the vegetative tissues, which are from the embryos are from these vegetative cells. So this is the whole process taking place in the tissue culture. Now we will see a brief flow of this tissue culture technique. This is the flow chart showing the plant tissue culture technique. Here you can see the flowchart clearly explained that first one, this is a preparation of medium, and this medium is taken in the test tubes, and the sterilization and autoclave method, everything is done in this medium. And other side, the plant is taken, that is the explant is taken from the plant, that is any small part from the root or the tip, some small amount of cells are collected from the plant, and here that explant is taken. And next one is this explant should be transferred into the medium. So next one is the step, you can see, that is a inoculation, that is under the sterilized condition. This explant, then now next it is incubated, and in this process the first formation is the callus formation, and after the callus formation then they are kept in the more amount of growth regulators medium. Then from that organogenesis organogenesis is done, that is formation of the small organs of the plant, and then root formation, and then transferred into a plants. And after small plantlets are reached, they are transferred to the glass house or the greenhouse, what we call, where they're very carefully they are we have to look after to that those plants, and if they are successful in growing in the plant freely, then it will be transferred into the field. And this is one process. And also other process is the somatic embryos, the somatic embryogenesis. The embryos are collected, and this embryo and these embryos are artificially seed preparation, and then plantlets are formed from these, and this is nothing but the germination. And here this is one process, and other one is these embryos are transferred into the medium, and again the culture takes place, the process is same, and at last we get the small plantlets. All these plantlets which we get from this callus is first they are transferred to the glass house and then to the field. This is the overall flow chart of the tissue culture technique. This is available in your textbook. Now let us look into the advantages of tissue culture. More number of plants can be produced in short time. Virus diseases can be prevented by producing virus-free plants from shoot tip cultures. Seedless plants can be multiplied. Female plants are selectively produced through tissue culture. Somatic hybrids can be raised by tissue culture where sexual hybridization is not possible. Tissue culture of medicinal plants produce high value of products of industrial and medicinal importance. So these are the some advantages of tissue culture. That is at the end we should get more number of plants in very short time. So from one cell we are getting more number of plantlets. That is, if the explant, if you take small amount of cells, from each cell we are getting one plant. So yield will be more. And also virus diseases can be prevented. Means we are making we are developing the plant without any pathogens, without affecting any pathogens. These plants are prevented, and these plants are happily cultured in the labs. These the seedless plants can be multiplied, and also this is not formed by the hybridization or so some seedless plants can be multiplied here. Female plants are selectively produced through tissue culture. So we can produce the same if you take callus, if you take the explant from the female plant, we get female plants only. Like that this culture is maintained. And somatic hybrids can be raised by tissue culture where sexual hybridization is not possible. Here no hybridization is possible in this technique. And culture of medicinal plants, because we need more number of medicinal plants, so the tissue culture is very useful for this purpose. On the basis of intermediate board examination point of view, I am giving the very short questions, very short answer questions and long answer questions. Usually long answer can come from this chapter, and also one very short answer also can come from this chapter. So let us learn these questions and make it as an assignment. Very short answer questions. What is meant by germplasm collection? Define totipotency and explant. Which part of the plant is best suited for making virus-free plants and why? And coming to long answer questions, if you're a botanist working in the area of plant breeding, describe the various steps that you will undertake to release a new variety. And other question, describe the tissue culture technique and what are the advantages of tissue culture over conventional method of plant breeding in crop improvement programs. These are the questions. You can note it as an assignment. That's all about the chapter strategies for enhancement in food production. And in the next class, we are going to learn the microbes in human welfare. This is all about in this chapter. Hope you understood. Please go through the textbook again. And also the essay answers, one essay answer will come from this lesson. So please study hard. And thank you for watching. All the best, children.